I'm Jonah Comstock, Editor-in-Chief at Pharma Forum. I'm here at JP Morgan 2024, actually at the Informa Biotech Showcase, and I'm joined by Xavier Duportet. He's the CEO of Eligo Biosciences. Thanks for joining me.
Well, thanks for inviting me.
So tell me about Eligo.
So we are a gene editing company, but not the typical gene editing company focusing on correcting human genes. Instead, we're focusing on addressing diseases that are triggered by the expression of bacterial genes, so genes from our microbiome. And so we've developed really a first-in-class modality that enables us to deliver into the microbial populations in our body, on the skin, in the gut, in tumors, specific DNA payloads that will express either CRISPR to knock out bacterial genes or base editor to genetically modify the genes in the microbiome, or actually therapeutic payloads to functionalize the microbiome and turn bacterial population into local drug factories.
Can I ask how you ended up in this area? I mean, it's so many layers deep, right? Already the microbiome was already sort of not as much lately, but originally kind of an obscure area, and then to do gene therapy on the microbiome. What's the story there?
So I was lucky to work during my PhD at MIT and together with a professor at Rockefeller University, Luciano Marafini, who is one of the co-founders of Intelia Therapeutics. And so they're doing gene editing for eukaryotic cells. And so while CRISPR was being discovered, and first application in human gene editing were appearing, we actually took the tangent and tried to see how we could use CRISPR and turn against the bacteria. And so we were the first one to demonstrate that if you deliver a nuclease into a bacteria and you guide the nuclease to create double-strand break in a chromosomal region of the bacteria do not have the same repair mechanism for the DNA, and so therefore it leads to the death of the bacteria. And so we had a completely novel mechanism of action that enables us to kill bacteria based on their genetic signature. And so microbiome was a nascent field at the time, and it started to appear that you had some very specific strains of bacteria that were associated to disease. At the same time, we all know that antibiotics are great to fight infections, but for more chronic diseases, you do not want to eradicate the whole microbiome. You want to have a much more precise action. And so this is when we started to think, oh, well, we could use this CRISPR technology to target the microbiome and have impact on a number of diseases where you want to have a very precise modification of this microbiome. And then, we delivered CRISPR, and then we delivered other base editor, other genes to be expressed. And so now we've just closed our Series B, a 30 million round led by Sanofi Ventures, to get into the clinic, our lead program, which is focusing on moderate to severe acne vulgaris. So it's going to be a topical gel where we're going to deliver CRISPR to the skin microbiome, and we're guiding the nuclease to target very specific genes within one bacterial species on the skin that we all have, but it is enriched in acne patient, and target the nuclease to specific pro-inflammatory genes to kill this subpopulation of bacteria that drive lesions on the skin.
Interesting. And I mean, is the applications for this technology potentially go beyond skin to
yeah, of course. So we're going on the skin first because we can go directly to patients. Actually, it's been 30 years that there's been no innovation in the acne space. So we're not focusing on the mild acne. We're really focusing where the unmet need is. So in moderate to severe, so it's when you have more than 40 lesions up to 150 lesions, and there's no treatment that really works for that. And it's 20 million people just in the US. It's a huge market, more than 10 billion. And so this is where we're going first to prove to have a very valuable asset after our first clinical trial, a phase 1b/2a, but also demonstrate that the platform can edit the microbiome. Then we can expand it to beyond skin. We actually have programs on skin, but also a program on tumor microbiome. 80% of our tumors are colonized by bacteria, and we know that these bacteria do trigger specific pro-tumorigenesis pathways in tumors or act as antagonists to drugs. And so there's a big, actually now it's getting quite hot about let's target very specifically the bacteria in the tumors to improve survival by removing these pro-inflammatory pro-tumorigenesis compounds in factors in the tumors. And the gut microbiome is also something that we're highly interested in. We've actually had a first pilot program in infectious disease, focusing on a bacteria that produce toxin in the gut. We brought it up to IND, and so that works really well. And we actually have a non-disclosed partnership with a top 10 pharma, focusing on a gut indication also. And so, what's fascinating about this field right now is that we're starting to have more and more of these cases and indications where specific genes from bacteria either on your skin, on your gut, in the tumors, in the lungs, even in the vagina, do drive disease. And so now it's about developing the right delivery vector to target this different microbiome, and then perform what is required. The CRISPR to knock out gene editing, to modify, to inactivate a gene, to inactivate a catalytic site in an enzyme of a bacteria, or produce biologics locally.
It's really crazy to imagine that you could have a topical gel and it's doing gene editing, not on you, but
on your microbiome.
Yeah. Is there any risk of, how do you target to get just the right bacteria, not the wrong bacteria, and to not get any of the person's cell? Is it different enough that that's not a concern?
Yeah, so we're using a specific delivery vector that is derived from bacteriophages. So there are viruses that target bacteria only, not human cells. It's the natural predator of bacteria. And so we're just using the envelope of these viruses, and we strip them out from their own genome, and instead we package our DNA payload with a CRISPR or the base editor. And so this is the first layer of selectivity that will only deliver the payload in bacteria and in the species that we're interested in. And then the second layer of selectivity comes from the CRISPR that will kill just the subpopulation that do carry the genes we're focusing on.
So you mentioned a big funding round led by Santa Fe, so that was just announced recently, right?
Yeah, it was in December.
And I think you guys announced a hire too, pretty recently.
We've hired many people. Right now the latest hire was our head of CMC, but no.
Yeah, I think that's what I was thinking of.
Okay.
So things are going well. You guys are getting some recognition. You're starting to get some attention. Has this been a good show for you?
Yeah, it's been actually quite an interesting show. We're actually meeting both investors, but also pharma. There's quite some interest now, and there's regained interest in the microbiome, I feel, and as to build V2 of the microbiome drugs, so much more precise. And I think really being at the intersection of gene editing and the microbiome enables us to address, provides to the gene editing field much more targets to be addressed. I think we got some really interesting meetings.
What's coming up for you guys?
So what's coming up? So basically now we're fully focused on execution, on getting this lead asset in acne vulgaris to the clinic as soon as possible. We should start trials in 25 to have readout patient data by the end of 25. And so this is our major focus for the company, and delivering on the partnership we have also.
To what extent does the gene therapy news that's focused on the human genome, the stuff that's going on with the regulation, with the FDA kind of becoming more accepting of it and building mechanisms, how much does that help you, in gene therapy, but in this different sort of area of it?
Yeah, so that's a good question. I think we're not facing the same challenges as human gene therapy, in the sense that we don't really off targets are not a really big deal for us. If you deliver in a bacteria you shouldn't deliver and you kill it, well, this is going to happen at extremely low frequency, and it's never going to be when you take antibiotics that kill all the bacteria, you don't really have major side effects. So off targets, which are a big deal in gene editing for eukaryotic cells, are not really such an issue for us. And so, but you're right, everything moving towards getting gene editing therapies, something more standard, is of course something great for us.
The rising tide, right?
Yeah.
You mentioned antibiotics, and maybe I'm completely off topic here, but is there potential for this kind of technology to have an impact on antimicrobial resistance and superbugs?
Yeah, so it can have. This is so we have some infectious disease programs, but we're only partnering them. We're not really developing them ourselves. I think we unfortunately, even if it's a major issue, we have, and given the current investment climate, developing a new solution to try to address antimicrobial resistance is something humanity needs, but unfortunately not very bankable.
Yeah.
And
I've heard
so,
that before.
so what we're trying to do is really actually leverage this platform first for really bankable application indications to really prove the potential of the platform. And of course, then it's likely, I mean, it does have application in antimicrobial resistance in AMR, but this is not where we're focusing most of our effort right now.
We probably are almost out of time. So anything sort of bigger picture, trend stuff you've been hearing or you're excited about for 24?
So I think, I mean, in our field, in the microbiome, what I'm quite excited about is that there's now regain of interest in the field. We're seeing some companies, actually other companies, funding again in the field. And with the advance of all the sequencing technologies, we're starting to see, I mean, it's much more credible right now. It's becoming much less a black box, and we start really to have really good targets for precision medicine of the microbiome, whether it's for us or for other microbiome companies. But I think the field is gonna, there was the hype then 5 years ago. Now it was not really, microbiome was not super sexy 1, 2, 3 years ago, but now with the much more solid science behind it that is often done by academics right now, the data generated is really good, and we're moving from correlation to causation, and this is what the field needed, and this is where we're going. And so I'm getting really excited for the years to come, because for us and for others now, the targets are going to be much more validated, and which will translate into higher rate of success for trials.
Yeah, which then translates to people taking it more seriously, more investment, all that stuff. Great. Well, thank you so much. I'll be definitely watching this space more closely.
Well, thank you.
I really appreciate learning a little more about it from you, Xavier.
Thank you. Enjoy the show.
You too.